Well, welcome and thanks for joining me today for the second episode of Season 3 of the Healthy Skeptic MD podcast. I'm Dr. Michael Hockman. This season, we will continue our focus or discussion of key common health decisions, and we're going to focus in particular on emerging health technologies. So today we're going to be discussing what, in my opinion, is perhaps the most exciting area of advancement in healthcare right now, and that is gene therapy. In fact, the Nobel Prize was awarded to two scientists this year, one from Berkeley, Jeniffer Doudna, and another French scientist, Emmanuelle Charpentier, who developed a tool called CRISPR, which is used to edit DNA, much like a word processor. It's a really amazing story, and there was a book about it by Walter Isaacson called The Codebreaker that I just finished. Really great. I would commend it for anyone interested in learning about that. But my guest today is Dr. Mark Walters. He's at the UCSF Benioff Children's Hospital in Oakland, and he and his team are actually using CRISPR technology to treat real patients with gene therapy, specifically sickle cell disease and thalassemia, a very noble cause. So it's great to hear his interview today. Please stick around for that. If you do find today's episode engaging, please make sure to subscribe wherever you listen to podcasts. And of course, on YouTube, please pass it along to friends, family, and colleagues who might be interested. And if you do have a chance, please consider leaving a review. Okay, so before we jump into our interview today with Dr. Walters, as always, let's start with a rundown of the health news for the week. And first, I want to start with Israel, where the update is that almost 60% of the population is now fully vaccinated against COVID-19. In comparison, here in the United States, that rate is only about 25%. In Israel, the case rates have dropped from a peak of about 8,000 per day in January down to just a couple hundred a day. And there's only 250 people right now in Israel who are hospitalized with COVID-19. And this is all despite the fact that gyms, restaurants, and theaters have been reopened for a while now, and they've actually lifted the ban on use of outdoor masks, which is relevant here in the United States, as I'm going to talk about in a moment. So the Israel example is really important because it does show what is possible if we can get this vaccine widely distributed in our population. So picking up on the Israel experience, my second news item I want to cover is the new emerging CDC recommendations about mask use, or the easing, I should say, of the mask use restrictions here in the United States in outdoor settings. Specifically, the CDC says that if you've been fully vaccinated for at least 2 weeks and you're alone or in small groups and you're outdoors, it is okay not to wear masks. This absolutely makes sense. The CDC does say that if you're in crowded areas in close proximity with others who are not vaccinated, you still should wear a mask. Now, a lot of my patients have been asking different scenarios and specific situations, and I think you can make yourself crazy trying to map out every scenario. But what I've been telling my patients is just to use common sense. If you're in a crowded setting, you're forced to be in close contact less than 6 feet with others who aren't vaccinated, it's probably still a good idea to use a mask. But the good news is that most of the time, if you're in a sparsely crowded area, you're not close to others, it is perfectly fine if you're outdoors not to wear a mask if you're fully vaccinated. The third news item I want to comment on this week is the CDC's recommendation to resume use of the Johnson & Johnson vaccine. It does include a warning for women in the age group of 18 to 49 about those blood clots. Now, overall, I agree with the CDC's recommendation to get the vaccine back on the market. But if I were a woman in that 18 to 49 age group, I would try to get the Pfizer and Moderna vaccine. Now, to be clear, even in that 18 to 49 age group among women, the risks are low. The rates of these clots is about 7 in a million. And according to the CDC calculations, the benefits of the vaccine in terms of preventing COVID outweigh that risk. But in my mind, if you are able to get the Moderna and Pfizer vaccine and you're a woman in this age category, why even bother taking the risk? So it seems like the intuitive thing to try to get the Pfizer and Moderna vaccine. And that's what I've been recommending for my female patients in the 18 to 49 age category. So that's it for the news summary this week. Let's jump into our interview with Dr. Walters. Well, thanks for joining me today, Dr. Mark Walters.
My pleasure. Thanks for having me.
So you are at UCSF Benioff Children's Hospital in Oakland. We're going to spend most of this podcast talking about your research work, but maybe you can just begin by telling us a little bit about your clinical practice there.
Sure, happy to. So I lead the blood and marrow transplant program at the hospital in Oakland. We're one of the hospitals covered under the University of California, San Francisco umbrella, one of the clinical sites. And so I'm also a professor of pediatrics in the School of Medicine at UCSF. So I matriculated to Oakland in 1999 specifically because of my interest in transplantation and the possibility of curing children with sickle cell disease, based in large part on the fact that there was a large comprehensive sickle cell clinic in Oakland. And not just sickle cell disease, but a related hemoglobin disorder, thalassemia.
We'll certainly talk more about sickle cell disease soon. What percentage of your time, out of curiosity, do you spend doing clinical practice versus research?
Currently, I spend about 20 to 25% of my time meeting with and caring for families affected by these diseases, and most of my time is spent in research and administration.
Okay, so we're going to talk today about gene therapy, gene editing. Some of these terms get a little hazy for the general public. Could you just begin by defining what is gene therapy and what is gene editing?
Sure. So what we're doing is collecting a person's own blood stem cells and then modifying them outside their body and then reinfusing them after an appropriate preparation after we've completed that modification. So gene therapy, also known as gene addition therapy, typically adds an extra healthy copy of the missing or defective gene, in which case in sickle cell disease is a beta globin gene. So the problem in sickle cell disease, as I'm sure many of you know, is that there's a change in the DNA sequence of that gene that alters the hemoglobin protein. So that once it's in circulation and has delivered its oxygen to the tissue and is on its way back to the lung to pick up more oxygen, it has this tendency to form these long chains, stiff rods that cause misshapen red blood cells to become stuck or adherent to the sides of the blood vessels. And that's when you block blood flow in a blood vessel, that can cause pain, it can cause organ damage. If it happens in the brain, it can cause a stroke, and so on and so on. So the idea here is that we can overcome that problem by adding an extra piece of DNA that contains a healthy copy of that defective gene. So that's gene addition or gene therapy. Gene editing is a completely different approach whereby we use these new tools to find, almost like a spell checker in your word processing program, to find a unique sequence in the DNA of the blood stem cell. And then once it's identified that proper sequence, it actually has another function that makes a clip, and it cuts the DNA molecule in half. And then when those ends are brought back together, as the cells should do, that often introduces a mistake. So that would inactivate a gene that you might want to inactivate. Or in our hands, what we're trying to do is actually correct the mutation of sickle cell disease. If it's going to work, and we have good reason to think it will, those cells now are reprogrammed just to make healthy hemoglobin. The sickle gene has been eliminated and replaced with a healthy gene. And so when those cells are returned, then the cells in circulation just make the regular kind of hemoglobin, and that's the way it exerts its beneficial effect.
Is it fair to say that gene editing is a type of gene therapy?
Yes, I think so. I mean, I think all of these fall under the category of gene or genomic therapies in that we are altering the DNA of the blood stem cell.
So gene modifications, editing is making the changes to the DNA, and then therapy is getting it into the body. And as you mentioned, you typically need some sort of vector, a viral vector, to distribute it to the cells and get it into the genome.
That's right.
So gene therapy, the idea has been around for actually a few decades now, but it got off to a slow start. There was an infamous case, a young man named Jesse Gelsinger, I believe, at Penn, who had a really bad reaction to an initial gene therapy attempt. Can you just tell us a little bit about the history of gene therapy and some of those initial stumbles?
Well, that was perhaps the most famous one, where the vector used in that case caused an immune reaction that caused a terrible inflammation in the liver and then it shut down, and unfortunately he died of that. I think what we learned from that is that the risk of that happening might have been predicted by some of the preclinical preparatory studies leading to the clinical trial. And I think the families understand who participate in these trials that this is a risky new therapy, and we don't understand all the side effects when we begin a clinical trial. But we really should inform families of the side effects, the potential side effects that we know about. So that was probably the most important error that's been corrected that was made in that particular trial.
Yeah, it was certainly a tragedy, but hopefully there were some lessons learned, and the idea itself did not hit a dead end. It was just a tragedy that unfortunately set the field back.
It did put a pause on gene therapy trials in this country for several years. The next iteration that I think is important is that there's another vector that we call retroviruses, and as it turns out, they have a tendency to go into the DNA actually near a cancer-causing gene. And this was tested in boys with a primary immune deficiency, which is the bubble boy disease. And unfortunately, because their immune systems were already so weakened and the virus landed near a cancer-activating gene, a number of them developed a form of leukemia as a consequence of using that particular vector. So what we learned was that we really shouldn't use that vector, particularly for primary immune deficiency. And when that shifted to using the lentiviral vector, which is the same one that we're using in sickle cell disease now, there have been no subsequent cases of cancer. So this is part of the learning that happens that unfortunately is necessary to first learn in human beings in the clinical trials. So that's why the informed consent, the transparency with the community of persons with the disorders that you're going to treat, how that's so terribly important to partner with the community as you develop these new therapies.
Yeah, it certainly seems like an iterative process, and you want to minimize as many of these bad outcomes, but sadly it's just part of the process, and you got to learn and move on when one of those bad things does happen. But they're fortunately happening less and less from what I hear.
That's right. But it's important to understand why they happen, and then re-engineer the treatment to overcome that defect.
So there was another really significant advance in this field less than a decade ago. The tool is called CRISPR. It was actually awarded the Nobel Prize for two of the key scientists who were involved in developing someone right near you, Jeniffer Doudna, who I believe you've worked with at Berkley, and another French scientist. Basically, they discovered this tool that bacteria use to chop viruses out of their DNA, and it so happened that same tool could be reprogrammed to edit genes. Maybe just say a word about the significance of that, particularly given that the Nobel Prize was awarded for it this past year.
Yeah, the significance is the versatility of the tools. The important discovery by Doudna and Charpentier is that you can swap out the guide RNA and then target virtually any sequence in the genome. There are a few constraints about how you target, but and it's incredibly efficient. So scientists have been able to modify the DNA in a targeted way, but it was a 1 in a million frequency, and now the efficiency, the frequency at which the targeting occurs is approaching 100%. Depending on what the particular target happens to be. So that was really the game changer, going from a very small fraction of genes actually being edited to all the genes being edited in the target cell population. The other important advance is that as this gets used more, the cost goes down. So as it turns out, it's becoming less and less expensive to be able to modify these tools so that you can use them for clinical application. And I suspect that will continue.
And as I understand it, also it's a lot easier. It's actually a lab tech in a clinical hospital with some training, but they don't need to have a PhD and multiple years of experience doing these experiments can actually make this work, if I understand it correctly.
That's right. College students, even high school students could pull this off.
So in this past January, the New England Journal published a case report of two cases. It was the first time in at least a number of years where gene therapy has been used successfully to, I think it's fair to say, cure or at least dramatically improve the condition of sickle cell disease and thalassemia. You actually wrote the editorial putting that new study in context. Tell us what happened for those two patients and how was CRISPR used to edit the DNA, fix the genetic problem, and dramatically improve their clinical condition.
Yeah, so these were two patients, one with sickle cell disease that I've just described, and another with thalassemia, which is another hemoglobin disorder. The problem there being that the beta globin gene is missing, so that it can't be produced. So those persons who inherit thalassemia need to be treated by transfusions very early in life, generally the first year or two of life, in order to survive. And then it's lifelong transfusions. So you can imagine that is cumbersome and expensive, and there are other side effects as well. So there's a condition that occurs naturally, a hereditary condition that we call hereditary persistence of fetal hemoglobin. So fetal hemoglobin is the type of hemoglobin that's made before a baby is born. It's in the womb, and it makes sense that you might need a different hemoglobin in that setting because obviously the baby in the womb isn't breathing air, it's getting its oxygen from the mother's circulation, from the mother's bloodstream. So this is a hemoglobin that binds oxygen more tightly than the adult hemoglobin. And then at birth, when that type of hemoglobin is no longer needed, it's turned off. Well, there's a hereditary condition whereby the fetal hemoglobin never turns off. And when it's inherited with either thalassemia or sickle cell disease, it makes both diseases much milder or even like trait, like not even having the condition. So the team and other scientists have reasoned that, well, if we could increase the fetal hemoglobin reliably, we might make these diseases milder. So what the New England Journal articles, one team from Boston Children's and the other team from a company called CRISPR Therapeutics, partnership with Vertex, they used, in one case, the CRISPR reagents to eliminate a repressor of that fetal hemoglobin. And what they did was they knocked out or eliminated that gene in the red blood cells, and that allowed the fetal hemoglobin to be expressed at a very high level, and in both diseases appeared to have a curative effect in that the hemoglobin increased such that transfusions were no longer needed, and in the sickle cell disease patient no longer had painful episodes that are the hallmark of the disease.
And to be clear, they made these changes on the bone marrow of these patients, and they actually had to do a bone marrow transplant to eliminate the regular bone marrow cells and then put in the genetically modified ones. Did I understand that correctly?
Yeah, that's right. In fact, they use one of the same drugs we use when we do a bone marrow transplant with using a brother or sister as the donor. So it is necessary to first destroy all the sickle cell or thalassemia producing blood cells with a very high dose chemotherapy drug, and then in that empty space repopulate it with the modified cells that then can regrow in the empty bone marrow space and then begin to make the fetal hemoglobin at a high level and mitigate the consequences of both disorders.
Okay, and you started to mention this before. Sickle cell, thalassemia are not pleasant conditions. You require blood transfusions. You have these pain crises with sickle cell disease. And after this genetic modification, gene therapy treatment was done, those improved dramatically. So tell us a little bit about how those patients did after their treatment.
Both in the most recent New England Journal study, but in a larger group of patients treated by gene therapy, sponsored by a company called Bluebird Bio, there's even more information about what happens after a successful treatment, which is that the painful episodes, the ones that are severe enough to cause to have to be treated in the hospital, are almost entirely eliminated, and that's the hallmark of the condition. The other problem is anemia. So the red blood cell characteristics almost normalize after a successful treatment. So they have a longer lifespan. The signs of their being broken down are eliminated from blood test results, and they feel better. So there have even been patient-reported outcome measurements using standardized instruments that test that show that they report less pain and fatigue. They generally feel better, and they sleep better. So they get better sleep. They're not being awakened at night because of discomfort and pain. So at several different levels, there appears to be a strong clinical benefit.
Is there any reason to think that the changes won't last, that the
Yeah, well,
old cells will come back?
yeah, there's good reason to think that it might not last forever, and uh, that's that's completely unknown. Now, so far, it's been durable through the 5 to 6 or even 7 years of follow-up that we have. But every stem cell in our bone marrow has a limited lifespan. And as we get older, we have fewer and fewer of them. We know that the diversity of cells that contribute to our blood um, diminishes with age. So if enough of those carrying the healthy copy of the right DNA fall out of the equation, then you might be left with anemia again. So, but that's going to take probably decades to learn.
So there's still a lot to be studied here. We've only done this on a handful of patients, but say the data hold up, it really proves to be a very encouraging option for many patients. I believe there's about 100,000 people in the United States who have sickle cell disease, millions more worldwide. Do you see this as a viable treatment option for that large number of patients?
No, I don't. And the reason why it's an intensive and right now expensive procedure, and even under the best of conditions, it takes about 6 months for the immune system to recover and reconstitute. So patients have to really put their life on hold for 6 to 12 months if they're going to go through something like this. And it's expensive, and our healthcare system, frankly, has a poor track record of supporting expensive therapies for all patients. So that's another hurdle that would need to be overcome. And technologically, it's challenging. So while we might be able to do it in a wealthy nation like this one, I'm not sure we could say the same in other countries in the world where resources are thinner. So no, we need to make significant advances in both the cost and the ease of delivering the therapy before it can be more widely utilized. But this is the proof of principle. And it's from this early step that we'll obviously definitely make improvements. So that's why it's so important.
Well, one of the critiques I've heard of gene therapy is that it could worsen disparities if it's the wealthy individuals and the well-resourced nations that have greater access to it. I don't myself see that as a reason to not move ahead, but I just think it's something we need to be thoughtful about as we do proceed.
Oh, I agree. I think health policy planning must go hand in hand with the scientific advances for us to have an impact on these disorders.
Now, in terms of the point about how do we make this a better treatment, how do we fine-tune it, how do we make it so that it would be cheaper and more realistic to treat a large number of people, I believe your lab is working on that. Maybe you could just tell us a little bit about some of the next iterations and tinkers that your team is planning.
Right. Well, so right now we do this manufacturing, if you will, of the cells of what we call the drug product outside the body. So those are done in tissue culture flasks. It's expensive. It's cumbersome, and so there are a lot of reasons to want to be able to deliver the therapy in the bone marrow where the cells reside. So one way we might begin to pursue that, and this is still theoretical, is that we might be able to package these tools, the CRISPR reagents, in what we call a nanoparticle. So it's a lipid spherical structure, small enough to inject, but big enough to contain the reagents. And once they're packaged, we might also put on that nanoparticle, let's say an antibody or a target that leads it to glom onto the stem cell where it is in the bone marrow. And then once it's taken into the stem cell, it could do its reaction, and then that particular stem cell would be corrected. Now you can imagine that the efficiency of that, getting even 10 or 20% of the stem cells modified by injecting a nanoparticle is a tall order. And it would need incredible targeting and efficiency. But that's the kind of thinking that's going on at the Innovative Genomics Institute at UC Berkeley that we're partnering with.
So it sounds like we've made a lot of progress, particularly with CRISPR in the last decade in the gene editing part. Seems like the next big hurdle is the best way to deliver those edited, modified cells where they need to be in the body.
There are some tricks in medicine and science that might help us from completely unrelated fields that we could apply to this particular problem in a unique way. So we always keep our ear to the ground as to what's going on in the rest of science and medicine, because there are tricks that we could make use of.
Is there an analogy to cancer treatment where you have to target chemotherapy just to the cancer without damaging other tissue? Is that an analogy too?
I think that's a fair analogy, yes.
Yeah. Okay, well, you're a pediatric hematologist, so you've been thinking a lot about sickle cell and thalassemia, but your mind can go in a lot of different places. You could envision there's genetic components to cancer and to heart disease and diabetes, many other conditions. Have you started to think about the possibilities of where gene editing, gene therapy could go for other conditions?
Right. Well, my area of expertise is in hematology, and there are lots of other hereditary hematological disorders where we understand the genetic basis for these. There are hundreds of different mutations that cause thalassemia, and in a perfect world, if we could ramp this up in a personalized medicine approach, we could create unique reagents for gene editing for each of those unique mutations that give rise to thalassemia.
And this may be a little out of scope, but any other major disease categories outside of hematology that come to mind for you?
Yeah, there's muscular dystrophy, where there's a problem with the reading frame of a particular protein in the muscle, that if you made the right edit, you could restore that reading frame and make a smaller but still functional protein that would overcome the problem in muscular dystrophy. So wherever there's a genetic defect that we've defined in a variety of different disorders, not just blood disorders, this is theoretically approachable.
Well, I have to say, I think this is the most exciting area of medicine. There's a lot of good things going on in medicine, but biotech right now, the changes and the opportunities over the next several years with gene editing, gene therapy, I can see some really big advances. But this is the Healthy Skeptic MD podcast, and of course we do have to ask some skeptical questions, and there are some important concerns. So maybe I could just end by asking you a couple of about some of those. So one of the big ones is in 2018, there was a Chinese scientist who went a little bit faster than the international community was ready for and edited embryos. So what's known as germline mutations that could be passed on. It was for a gene it makes people susceptible to HIV. So, there's no reason to think per se that there'd be any major problem with that, but it really is opening up a Pandora's box once you start changing germ cells that can be inherited, and that's where you get designer babies that can change the trajectory of evolution. Just any thoughts about that, and how do we prevent that, and are we ready to start moving in the germline direction?
No, I don't think we are. I think we're still learning about. So I've been focused on in a very optimistic way the on-target capabilities of these reagents, but like everything in life, there are off-target effects, and so while this is precision designed, it's not necessarily precision all the time. So applying the CRISPR reagents in germline without understanding how to monitor for and avoid off-target editing in genes that would be terribly important in the developing embryo and with regard to cancer risk is completely unwarranted and unethical.
So that's not even just an ethical concern. That's a pure scientific concern that there could be some real safety problem with doing that.
Yeah, there are some profound safety concerns, and as physicians, we're first
do no harm. So
that's just not only unethical. It should be condemned by the scientific and medical community in the way that it was. So, but even in the way that we're applying this new technology carries significant risk, and we're still learning about that risk. So, and I think that was made public recently when not one, but now a second case of acute myelogenous leukemia developed in a sickle cell disease patient who was treated in a gene therapy trial. The excitement is based on real evidence that these therapies have therapeutic value, that the patients appear for all intents and purposes to be cured. But how do we select which one? And it's going to really come down to which one is the least risky. So we need to understand the risks involved much, much better than we do today before we can begin to prioritize where we go in the future.
Well, I really appreciate your saying that, because while I think there's huge opportunity in this field, as you said, this is not for the faint of heart. There are some bad things that can happen if it's not done safely, methodically, and we don't learn from mistakes. Anybody who's getting themselves involved in gene editing, as you said, right now there's so much we don't know that there really is a risk, and it only makes sense to do it if you have a disease severe enough to justify that risk.
Right. Well, that's true.
So one theme throughout this podcast is a lot of new technologies. There's a lot of excitement, but they don't. The progress is slower in terms of leading to benefit to real patients as quickly as people expect. Why is it that this field might not progress over the next 10 years as quickly as many hoped?
Having been cured, it's fantastic. There's nothing better than not having to take medicines anymore and not having to experience the side effects and to having an extended lifespan. But it's at a cost, and financial cost, and long-term toxicity costs, and all of those have to be understood and weighed before going forward.
Well, I've learned a lot from your perspective on this. To summarize, it seems like there's been a big advance in terms of gene editing, in particular in the last decade. We're learning more and more about the genes that cause disease, which ones need to be modified. A big barrier right now is delivering those genetically modified cells where they're supposed to be within the body at the right time. And there's still significant safety concerns. To do autologous bone marrow transplant is not a small procedure, and these delivery mechanisms have their downsides. So there's some still important barriers that need to be overcome, but tremendous possibilities. And then finally, there are some ethical concerns as we move ahead, unintended consequences, and once we move into the germline and affect future generations that need to be thought through. And then I guess the very last thing would be the resource and the allocation issue that you mentioned, that how do we make sure that everyone benefits from these opportunities? A really, really fun field, I'm sure, to be in. Any predictions for how the next 10 years are going to go in this area?
I've looked in my crystal ball and I've come up blank. All I can say is I think it's going to be exciting and it's going to move quickly, but I can't predict how.
Yeah, well, I think many people are optimistic, myself included, but you just never know how these things are going to turn out. But it's great that we now do have at least 2 patients and several more now, like we'll hear about soon, who have been cured of really bad conditions, sickle cell disease and thalassemia, or at least greatly helped by these genetic modifications. So thank you so much for joining me today.
My pleasure, Michael. Thanks for having me.
And if you found today's episode engaging, please do subscribe to the Healthy Skeptic MD wherever you listen to podcasts. Leave a review if you have a chance, and pass it along to others who might be interested. Thank you.